So moving on to traits, which are considered the phenotype, so the outward expression of our genes, but it gets more complex, right? So by now you should understand that human traits, even the very simple single gene traits that we often use as examples of Mendelian traits, are more complex than your typical Mendelian traits, right? Human traits don't fit the pattern that pea plant traits fit into. And so what I want you to get from the previous activity is to realize that a small number of traits are influenced primarily by a single gene. However, most of the time, even those single gene traits are really influenced by multiple genes, as in the case of eye color. And oftentimes, a single gene can influence multiple traits. For example, some genes involved in eye color can also influence hair and skin color. So let's look at one of those simple single gene traits. So bitter taste. Humans have about 30 genes that code for different bitter taste receptors. Why is that important? Well, I want you to realize that multiple genes code for similar functions. That's going to be important. And why do we have a bitter taste gene? Well, many naturally occurring bitter substances are toxins, so it's thought that the ability to discern bitter tastes may have allowed humans to avoid eating poisonous plants. And so genetic variation can be beneficial for survival. Why don't we all have the ability to taste, a particular substance? Well, the website goes over the advantage of being heterozygous for PTC specifically, in that you might be able to identify even more bitter taste if you're heterozygous. Okay, so looking at the PTC taste gene specifically. So there is an inherited component that influences how we taste PTC, or one particular type of bitter compound. The ability to taste it or not taste it is conveyed by a single gene that codes for a taste receptor on the tongue, and remember that receptor is a protein. And there are two common forms or alleles of the PTC gene. So there is a taster and a non-taster. And each allele codes for a bitter taste receptor protein with a slightly different shape. So the shape of the receptor protein will determine how strongly it can bind to PTC. On average, about 75% of people can taste it, while 25% cannot. So traits influenced mainly by one gene usually have a predictable pattern of inheritance. So this is usually a gene that we use to provide an example of Mendelian inheritance. But I want you to know that most traits have a complex, unpredictable inheritance pattern. So although PTC sensitivity is often used as an example of a simple Mendelian trait with dominant inheritance, tasters vary greatly in their sensitivity to PTC. So there's actually an incomplete dominance picture, or partial dominance, or semi-dominance, or intermediate inheritance. And this occurs when the phenotype of a heterozygous genotype, so one with a dominant allele and one with a recessive allele, is distinct from and often intermediate to the phenotypes of the homozygous genotypes. So if you inherit, if you look at Dad, he has all the tasting receptors and he can taste because he has the dominant receptor. If you look at Mom, she has all the non-tasting receptors, so she cannot taste it. But then the kids are heterozygous, and so they actually can taste it, but not as strong as Dad. And that's not the only thing that influences the phenotype. So while the gene is estimated to have 85% of the total influence over whether someone is a taster or a non-taster, There are other things that affect this ability. So, one is the environment. Having a dry mouth makes it more difficult to taste PTC. Also, what you ate or drank before sampling the PTC paper might affect the tasting ability. Okay, so now we have an incomplete dominance, so it's a little more complex than your typical Mendelian trait, right? And you have an environment that influences the phenotype, which goes against the Mendelian traits. Also, what you ate or drank before sampling, or also how old you are affects it. So your genetically predisposition to tasting PTC changes as you get older. Some people find that they can taste PTC on some days and not on others too. So your age, time, what you eat or drink, all of those things influence your phenotype or your trait, even though this is typically considered a genetically determined trait. So what you will see over and over in humans is nothing is genetically determined. The other complication that we see is even though there are two common alleles for PTC tasting, there are multiple uncommon alleles. And so imagine if we were looking at a fatal disease and we were trying to predict someone's outcome. And we are only considering the Mendelian inheritance of the two common traits or the two common genes. We'd be really surprised if we just excluded all of those uncommon genotypes. And that's what you see over and over as we look for common genetic mutations associated with diseases, that there's not just two, there's so many variations. So by now you should understand that human traits are very rarely fit into the Mendelian trait category. Our traits are difficult to measure, they change over time, they're influenced by multiple genes and environmental factors. Most traits are continuous rather than dimorphic. So think of, this being the example of dimorphic is like yellow and green, and continuous is like the color spectrum, right? We can have those categories that we make, but oftentimes they're, of course, categories that we just made up. Dominance is less predictable. Most genes have more than two allelic variants, and one gene can influence multiple traits, and also the environment can change your genotype, right? So we didn't even talk about how the environment mutates your genotype. So in humans, genotype influences but does not determine phenotype.